So this is a paper video about work published in the journal Brain in 2024, in which we compared single value scores of memory brain activity between Alzheimer's patients and other clinical groups. So this is the author list of this paper, and this paper was actually the follow-up to an earlier study in which we developed and validated summary statistics of memory-related fMRI activity as potential biomarkers for neurocognitive aging. In this study, we recruited young and older adults, and they were shown images which were either new to them at the time of presentation, here called novel images, as well as images which they had seen before a couple of times, here called master images. So this was the encoding session of our memory experiment, and this allowed us to infer on effects of novelty processing. Then around 1 hour later, subjects were shown all the images from the encoding session, now called old images, as well as a couple of new images, which they had not seen before. And their task was to rate all those images on a 5-point scale, depending on how well they believe to remember them from the encoding session. So this was the retrieval session of our memory experiment, which allowed us to infer on effects of successful memory encoding. In the original study, we either compared brain activity during novel images versus master images in order to infer on effects of novelty processing and then compare those between young and older adults, or we compared later remembered with later forgotten items in order to infer on subsequent memory effects and compare those between young and older adults. Based on those observations, we then introduced two fMRI biomarker scores, which are either quantifying the deviation of a given older adult's brain activity from that seen in young subjects, this is called the FATE score, or quantifying similarities of activations and deactivations between an older adult and the reference sample of young subjects. This is called the SAME score. For the present paper, we applied the exact same methodology, so same fMRI scanning, same statistical analysis to the DELCODE study, which is run across several German centers for neurodegenerative diseases, or DZNE in short. And from all the subjects in the study, we only took those with complete fMRI data. This gave us 128 healthy controls, individuals with subjective cognitive decline, SCD, patients with mild cognitive impairment, MCI, patients with Alzheimer's disease, AD, and relatives of Alzheimer's patients. So that in total, we have a clinical cohort with a sample size of 468 subjects. The participant groups are from the same age range, so all between 60 and 90 years old, but they are obviously differing on other characteristics, such as mini-mental status exam or the R-beta 40-40 ratio. So what did we find when calculating the scores for the subjects in our new study? The single value scores are obviously differing between participant groups in the sense that the memory scores allow to distinguish MCI and AD patients from all the other groups, whereas the novelty scores allow us to distinguish MCI and AD from each other. So we also calculated correlations of the single value scores with other variables after removing effects of participant group. And in this way, we found partial correlations between the scores and chronological age, memory performance, neuropsychological testing results, and neuropathology, at least in part. The participant groups are, of course, also differing as expected with respect to the ApoE genotype. We have a lot more E4 carriers in MCI and AD patients. However, there are no effects of ApoE genotypes on the scores themselves, except for in relatives of Alzheimer's patients, where we have significant differences between E3 homozygotes and E4 carriers. There are also no effects of amyloid status on the scores, except for in individuals with SCD, where we have significant differences between amyloid positive and amyloid negative subjects. So these are some results from the paper. And if you want to know more, don't hesitate to check it out, which is available open access from the publisher's website. Thank you very much.